Good afternoon. Myself Vaishali Sonawane from the Department of Microbiology. I am working in MVP's S.V.K.T Art Science and Commerce College, Devlali Camp. The topic for today's lecture is hepatitis A virus, and in this particular topic is meant for TYBSC microbiology student. So let's begin with our lecture. Before beginning with our lecture, we'll have a brief recap of what we have discussed in our last lecture. So in last lecture, we have discussed about the general characteristics of viruses, what are the basic characteristics of viruses, how you can differentiate viruses from other organisms. Along with that, we have also gone through the classification of viruses. Today's session agenda is firstly talking about hepatitis A virus, but before that we are going to discuss some of the important terminologies that are concerned with hepatitis viruses or infections that are associated with liver. Then we are going to move on to the morphological characteristics of hepatitis A virus. Then we are going to see the differences between different types of hepatitis virus, followed up by the mode of transmission of virus, symptoms associated with hepatitis A infection, and lastly we are going to see the prevention and control of hepatitis A infections. So these are the learning objectives for our today's lecture. So our main focus is on hepatitis A virus, and under that we are going to discuss all aspects of hepatitis A virus. That is, we are going to see the common terminologies, we are going to see the symptoms, we are going to see the difference between different types of viruses. Then we are going to look on to the mode of transmission of hepatitis A virus, disease, symptom, pathogenesis, and along with that at last we are going to see the prevention and control of hepatitis A viral infection. So first let's see what is a viral hepatitis. So the term hepatitis, we all know that hepatitis is basically a systemic disease in which liver is generally hampered. Liver is the main target site of hepatitis virus, which leads to inflammation of liver, and this inflammation of liver is generally caused because of the various viruses, various types of hepatitis viruses like hepatitis A, B, C, E. There are different types of hepatitis virus, and all of them can contribute to the viral hepatitis. In addition to hepatitis virus, but like not only hepatitis virus, but other group of virus like your cytomegalovirus, Epstein-Barr virus, yellow fever virus, all these are also associated with the inflammation of liver or hepatic conditions. So let's begin with the basic terminologies which we are going to use throughout our presentation and which are associated with hepatitis infection. So the very first term is the hepatitis. Hepatitis is nothing but it is an inflammation of liver. So in any condition, if there is an inflammation of liver, it is commonly termed as your hepatitis. Acute viral hepatitis. Acute viral hepatitis is the hepatitis in which the symptoms of hepatitis last for less than 6 months. So it is not that much severe type of hepatitis. Then next is acute hepatic failure. Acute hepatic failure is the serious complication that occurs in hepatitis infection, and which can lead to the liver failure, loss of function of liver, there is a severe damage of liver. Then next is the chronic hepatitis. Chronic hepatitis is the hepatitis in which the symptoms last for a period of 6 months or more than that of your 6th month. Cirrhosis. Cirrhosis is the condition in which there is a complete damage of liver, and therefore liver transplantation or replacement of liver is required. Then fulminant hepatitis. In this particular hepatitis, there is an because of damage to the liver cell, there is failure of liver. So these are the common terminologies that are associated with the hepatitis. So let's see different types of hepatitis virus. So this is a table showing different types of hepatitis virus and the common difference that are present in these all hepatitis virus. So let's see, when we talk about hepatitis A virus, so the agent is of this particular infection that is caused is hepatitis A virus. Hepatitis A virus is single-stranded RNA virus, and it is an unenveloped or non-enveloped virus. Moving on to hepatitis B virus, it is double-stranded DNA virus which is an enveloped. Hepatitis C virus is single-stranded RNA enveloped virus. Hepatitis D is again single-stranded RNA enveloped virus, and hepatitis E, it is single-stranded non-enveloped virus. So, in among all these viruses, you might have noticed that it is only your hepatitis B virus which is a double-stranded DNA virus. Then talking about mode of transmission of all these hepatitis virus, hepatitis A virus is majorly transmitted via oral fecal route. Hepatitis B by parental route, vertical transmission is there, sexual transmission is there. Hepatitis C is again parental transmission, hepatitis D is again parental transmission, and hepatitis A, similar to that of your hepatitis E, similar to that of your hepatitis A, is fecal oral route. So, moving on to the next is the age group of humans that are infected. So, hepatitis A infection generally occurs in case of small children. Hepatitis B can infect children as well as adults. So, any age group can get infected with hepatitis B viral infection. Talking about hepatitis C, it is restricted to adults, hepatitis D to any age group of people, hepatitis E to generally young adults. Talking about the carrier state, hepatitis A doesn't have a carrier state, whereas your hepatitis B, C generally has a carrier stage. So, carrier can transmit the viruses without showing symptoms. So, in case of B and C, there is a presence of carrier state. Then talking about the incubation period for all these viruses, for hepatitis A, it is 10 to 50 days. For hepatitis B, it is 50 to 180 days. For hepatitis C, it is 40 to 120 days. For hepatitis D, it is 2 to 12 weeks. And for hepatitis A, it is 2 to 9 weeks. Talking about the severity of infection that is caused by hepatitis virus. So, hepatitis A virus doesn't cause a very chronic infection, hepatitis A as well as hepatitis E virus, but hepatitis B, C and D can cause the chronic infection. Talking about the immunoglobulins and vaccines, it is there for hepatitis A, B and D. So, this is an overview of difference between the different types of hepatitis A virus. So, let us begin with the actual virus which we are going to discuss in our lecture is hepatitis A virus. This hepatitis A virus was previously or formerly called as infectious hepatitis or epidemic jaundice is the infection that is caused by your hepatitis A virus. This hepatitis virus, the common symptoms which are associated with hepatitis A viral infection is your fever, chill, nausea, vomiting. These are the common symptoms that are associated with this particular organism. The disease generally is a self-limiting. The infection that is caused by this organism is a self-limiting. So, after some period of some weeks, the disease itself gets cured. The organism, this particular virus, just now we have seen that it is your RNA virus, and as a general characteristics of viruses there, this organism it requires a living host for its growth and development as well as replication. Talking about the morphology of this organism, hepatitis A virus, it belongs to Picornaviridae family. So, it is a small virus. It ranges in the diameter, it has a diameter of 27 nanometer. It is non-enveloped virus with an icosahedral symmetry. It is RNA virus which is single-stranded RNA. Then as I told you that it belongs to Picornaviridae and there is presence of only one serotype of this particular virus. So, these are the common morphological characteristics of hepatitis A virus. So, this is the diagrammatic representation of hepatitis A virus. You can see there is a presence of capsid, the organism. This virus is non-enveloped, so there is envelope is not present. So, the external most part is the capsid. There is a presence of single stranded RNA and associated proteins are there. Now, talking about the host factor, which plays important role in the infection of this hepatitis A virus. The very first important factor, host factor, is the age factor. Age factor generally, hepatitis A virus, it infects the small children. Now, basically thing is what, this particular organism is transmitted via fecal oral route, and most of the kids, small infants, are not able to maintain their proper hygienic condition. So, that age group of children are more prone to that particular infection, or they are more susceptible to that infection. Second is the sex of the person. So, this infection occurs equally in case of males as well as in case of female. Talking about immunity, once the person is infected with this infection, immunity lasts for very long period of time. And along with that, in any case of infection, either it is bacterial, fungal, viral, any infection, if a person is having a poor immunity, in that case, that particular person is more susceptible to large number of infection. Then who is at risk? So, when we talk about hepatitis A infection, who is generally at risk? So, let us see, generally this particular virus or this infection generally occurs in poor sanitation. So, if a person is not maintaining proper hygienic condition, or the place where that person is living, or where that community is living, if proper hygienic condition is not there, poor sanitation is there, so that can that category of people are at higher risk. Apart from that, lack of safe water. So, if clean water is not available for drinking, so that can lead to this infection. The people living in household with infected persons. So, since this particular infection is transmitted via fecal oral route, so if a person is living with another person who is infected with this particular viral infection, so even that person is more prone to the similar type of infection. Again, talking about sexual mode of transmission. So, if if relation is done with a infected person, so even that can prone to the transmission of such diseases. Use of recreational drugs. These are the common things or common category of people who are at major risk. And the last one is those people who are traveling to the epidemic area or endemic area, or where hepatitis A infection is taking place, or which has occurred without being vaccinated. So, that even that particular person is prone to hepatitis A infection. So, all the people who are facing this particular type of factors, or who are residing in all these factors, along with these factors, are generally prone to hepatitis A infection. Talking about mode of transmission. So, since beginning with our lecture, we are talking about the mode of transmission. So, the mode of transmission of this particular virus is via fecal oral route. So, incubation period is 10 to 15 days, and fecal oral route, along with fecal oral route, other routes are also there, but the fecal oral route is the most predominant route by which this particular organism is transmitted. So, if a person is having contaminated water, or the water which is contaminated, which is having this particular virus, is more prone to the infection. Then parental route is quite rare as compared to that of fecal oral route, but transmission can occur. Sexual transmission, again it is rare, but it can occur. So, let's see the pathogenesis of hepatitis A virus. In this particular image, you can see that the infected liver cell is there and the organism is released via this particular liver cell. Now, when we talk about pathogenesis, in our previous slide, we have seen the mode of transmission of this particular virus. So, basically mode of transmission is via fecal oral route. Now, when this organism enters into the intestine via oral fecal route, via intestine, the organism along with the circulatory system, it is absorbed along with nutrients, it makes it move towards the circulatory system, and from that circulatory system, the organism enters into the liver cell. Now, when this organism enters inside the liver cell, it grows there, which leads to the liver damage or necrosis of liver cells. Now, when the organisms is growing in the liver, at that time whatever bile juice is there or bile secretion is there, along with bile secretion, that virus again enters into the gastrointestinal system. It enters into the intestine, and from there via fecal matter the organism is released outside the body of that particular patient. Now, if that contaminated fecal matter again contaminates the drinking water, in that case the organism can move from infected person to healthy one. So, this is how the organism moves from one person to another person via fecal oral route, and basically it damages the gastrointestinal system, along with that it also damages the liver. Now, let's see what are the common symptoms that are associated with hepatitis. Now, when we talk about hepatitis, hepatitis is actually the infection of liver, and most of the time the symptoms are more or less similar in case of all hepatitis infection. So, the very first symptom is the tiredness. The person feels tired, the person feels exhausted. Then there is a loss of appetite which leads to nausea. There is a fever, since the organism enters into the circulatory system, that leads to the fever. There is a stomach ache, then which is followed up by diarrhea, dark urine, yellowing of skin, yellowing of eyes. That is the major symptom that is associated with hepatitis. Moving on to the diagnosis, how we can diagnose this particular organism. So, for diagnosis, you can go for a serological analysis, you can go for microscopic examination, you can go for observation of this sample via in fecal matter, or you can use a blood sample for serological analysis. So, firstly you can demonstrate this virus from freshly stool sample, fecal matter, or in the blood or bile by using electron microscope. Then you can go for virus isolation and cultivation of virus in cell line, or you can cultivate this virus in the embryonic egg. Then talking about serological analysis, you can go for ELISA, in which you can detect the presence of antibodies against this antigen. Along with serological analysis, you can also go for biochemical test of blood sample. So, in case of liver infection or in case of hepatitis, there is an increase in alanine aminotransferase enzyme. So, you can detect its level. If it is high, it indicates that infection is there. There is an increase in bilirubin and proteins. Then for molecular diagnosis, you can surely go for PCR, so which can help you in amplifying a small fragment of DNA if at all it is there. So, by using all these techniques, you can identify whether the person is suffering from hepatitis A infection or not. This is the chart showing a time course of hepatitis A infection. So, in that you can see actually the virus enters into, the virus is released in the fecal matter as well as in the circulatory system after 2 weeks of infection. So, that much time period is required for establishment of that virus. Along with that, there is an increase in transaminases. So, you can see after 4 weeks of infection, there is a slight increase in rapid increase in your alanine aminotransferase enzyme, and symptoms begins after a 3 to 4 week of infection. Along with that, as a response to that antigen, initial IgM types of antibodies are formed in the beginning of infection. that is after 3 to 3.5 weeks of infection. But later on more specific IgG types of antibodies are formed. So, this is a time course of hepatitis A infection. Now, let us talk about how we can prevent the occurrence of hepatitis A infection. So, you can always prevent this infection. Now, as we have seen that mode of transmission is oral fecal route. So, any type of infection, either it is caused by bacteria, virus or any other different type of virus, if its mode of transmission is oral fecal route, the best way to prevent that infection is maintenance of proper hygienic condition, along with that maintenance of proper sanitation. That is the key for preventing this particular any infection which is transmitted via oral fecal route. So, even for hepatitis A, that is the major way by which you can prevent the occurrence of hepatitis A infection. Then secondly, there is a passive immunization of human immunoglobulin that can be given for the people who are at risk to avoid hepatitis A infection. Then there is active immunization is also there, and along with that several inactivated or live vaccines are there which can also help in avoiding hepatitis A infection. Talking about treatment. Now, when we talk about hepatitis A infection is generally a self-limiting. After some period of time, the disease itself stops, and therefore in case of hepatitis A virus, there is no specific treatment, no specific drug is available to treat this particular infection. The best way to avoid this, to prevent this infection is proper diet and a long rest, which is required for getting cured from hepatitis A infection. Generally, as I told you, that body itself gets cleared with the hepatitis A infection, and if at all your liver is damaged, liver is self-healed within a period of 6 months. So, this is all about the treatment and prevention of hepatitis A virus. So, let us summarize what we have seen in our today's lecture. So, in our today's lecture, we have gone through the morphological characteristics of hepatitis virus, followed up by the different types of hepatitis A virus, different characters of hepatitis viruses. Then we have seen mode of transmission of hepatitis A virus, pathogenesis of hepatitis A virus, and we have also gone through the symptoms, diagnosis, prevention and treatment of hepatitis A virus. So, lastly, we will have a assignment. So, there are 2 questions for your assignment. One is that write a note on pathogenesis of hepatitis A virus, and secondly, you are supposed to find out the differences between hepatitis A and hepatitis B virus. So, let us have a recap of what we are going to discuss in our next lecture. So, in our next lecture, we are going to discuss on another virus, that is Japanese encephalitis virus. So, Japanese encephalitis virus is nothing but it is basically the virus which infects the nervous system. From the name itself, you might have got an idea, since encephalitis there. Encephalitis is what? It is an inflammation of brain. So, this virus generally infects the nervous system. Under that, we are going to see the morphological characteristics of this virus. We are going to see mode of transmission, symptoms, diagnosis, prevention and control of the infection that is caused by Japanese encephalitis virus. So, thank you for today.